Welcome back to Particle Pharma. In last session, we have already got the knowledge regarding some of the organelles of cell. In today's session, I will be going through remaining organelles. So let me continue with peroxisome. It is an oxidative organelle covered by lipid bilayer membrane, which is selective in nature, or like that of the cell membrane, which is also an lipid bilayer structure, as that of the nucleus and mitochondria. These are also covered by two membranes, whereas it contains the enzyme catalases and oxidases and produces hydrogen peroxide. So this hydrogen peroxide, it will get converted into a water. So this hydrogen and oxygen, it will form an hydrogen peroxide, that is H2O2, and will get converted into water through a step of detoxification in the presence of enzyme catalases, as this hydrogen peroxide is an harmful substances. It is an harmful substance for the body. And they are also similar to lysosome, just like that, and formed by self-replication. So this smooth endoplasmic reticulum or endoplasmic reticulum, which is having cis phase and trans phase, through cis phase they will enter and through trans phase they will leave. The functions of peroxisomes are it breaks down fatty acids and form membranes and helps in respiration, and which we have already seen the conversion of hydrogen peroxide into a water, and it also helps in little amount of biosynthesis, I can say, not complete. So let me go to other subcellular organelle of the cell, that is vacuole. It is also an important subcellular organelle, which are membrane bound sacs, means they are covered by a membrane, single membrane, and it is having many functions such as it removes waste and helps in autophagy, cell death like that of lysosomes, and maintains acidic internal pH and internal hydrostatic or turgor pressure, whereas it performs exocytosis and endocytosis and store food, variety of nutrients and waste products. It contains water, organic and inorganic compounds and enzymes like that of the other subcellular organelles. Moving on to the other subcellular organelle such as centriole, which we have already seen in the brief discussion of the cell or terminologies of cell. So let me start with the description of centrioles. The region of centriole means the pair of centrioles which are arranged perpendicular to each other, and that region is known as centrosome, whereas the arrangement they are arranged perpendicular to each other, now that arrangement is known as diplosomes. So these are the microtubules. They are arranged in scaffold in anti-clockwise arrangement, and it has three parts. In the first part is the nine pair of or bundles of microtubules will be there, which are arranged right angle to each other. So that arrangement is known as diplosomes, whereas the first part or the distal part is the microtubules, which are lying away from the center. So we can easily count 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 pair of microtubules are there. Once again, I am showing it to you. 1, 1, 2, 3, 4, 5, 6, 7, 8, and 9. So these are the 9 pair of microtubules which are arranged perpendicular to each other and are away from the center, which we have seen it. So this is the first part, and the second part is the central part of the central core part. Okay, this is the central core part is there, on which microtubules are attached, and it stabilizes the scaffold. The last or cart wheel, so on which microtubules which are attached, that cart wheel, these are the filaments, they are radiating from the center. These are the filaments that they are radiating from the center. So these are the three parts. First are microtubules, second is central core part, third is filaments, or last cartwheel part. So microtubules are made up of tubulin proteins, which we have already seen. and they can get duplicated in S phase, that is in mitosis, and form the daughter centrioles. And these centrioles are converting into basal body. This basal body, it is present at the base of the cilia or flagella. Basal body is there, they are present at the base of the cilia or flagella. And they are also helps in cell division, which separates the chromosome. So they are having a scaffold of microtubules which are arranged in anticlockwise arrangement. Let me go to other subcellular organelle, that is cytoskeleton, which is helping in maintaining shape and structure of the cell. They are of three types: actin filament, microtubules, and intermediate filaments, actin filament or microfilament. So they are forming a bundle. See, they are forming a bundle, and it helps in determining the shape of the cell and helps in muscle contraction. Next is microtubules. These are the hollow rods. I can say microtubules or tubulin proteins. These are the hollow rods. They can easily assemble and disassemble. They are also assisting in daughter chromosome movement during mitosis to form a new daughter cell, and it provides support and shape. Last one is intermediate filament. These are the stable structure or mesh-like structure which holds a nucleus on its position and gives the elastic properties to cell, whereas it also try to uphold the other subcellular organelles like that of the nucleus. Moving on to the other or the last subcellular organelle, that is nucleus. Nucleus, they are lying in the center of the cell most of the time, which is a double layer organelle which is porous in nature, allows only some of the substances to pass through it, and they are in continuous with the endoplasmic reticulum. Sometimes the endoplasmic reticulum are studded with ribosomes, sometimes they are not studded with ribosomes. Those which are present with ribosomes are known as rough endoplasmic reticulum, and those which are without ribosomes are known as smooth endoplasmic reticulum. Its components are nucleoplasm or karyoplasm, which is present inside the nucleus, which is a semi-aqueous material which contains salt, enzymes, and molecules. It protects the nucleus, chromosomes, and maintains the shape of the nucleus. Next is nucleolus. The center part is nucleolus, which is present inside the nucleus. It is composed of proteins and RNA, and it is a site of transcription, and it helps in the processing of ribosomal gene. Next is the chromosome or the genetic material of an organism. I can say these are the thread-like structure, consist of DNA and histone proteins. These histone proteins are of four types, which we have already seen: H2A, H2B, H3, H4. On histone proteins, the DNA is coiled. And next part is chromosomes. Chromosomes are having three important parts. The first is kinetochore or the center part. The poles are known as telomere and the arms are known as chromatids. So this chromosome is helping in protein synthesis, cell division, and it stores the cell hereditary material which transforms from one generation to another generation, and RNA transcription. So by this I have completed most of the subcellular organelles. If you have any queries, try to contact us through various platforms. Thank you.